I hope you see. Can you see the screen? Yes, no?
Yes, we can.
Yes.
Good. And try to show me your face in Exchange. So the first thing is this. Well, I found this Pubmed training toolkit, which is kind of the material so people can teach. There are some videos here. 1 minute video for that is something that we should check, and we should consider this part of our content. I mean, it doesn't make sense that I make a video if the official people already make the video and they have more time and they know better. But the other thing that we can take is this one. I like it. This one is tip for using Pubmed is a PDF. It's kind of the thing that we should print and author last name and initial. And well, you can see here, and there is some link like PMC and this one. It will give you a link to the help, which is this help. And as you see, updated last Monday. And here they don't say anything about address, which is what I was looking for. But there are some interesting, like, there's a point here that I was wanting to see. What was one of the things that I wanted to see? When we say look for, Brown University, several people did, not everybody. You have to say and with big letters, no? The combining search, what is the Boolean operator? This one, the Boolean operation. And yeah, that was one of the things that I wanted to see. The dates can be specified like this. You have a range like start of 2010 until the end of 2010. In the last 5 years, you can specify the range like that. And the other thing that I wanted to show you is here. No, you have an option that can be interesting for you. Create an email alert. If you are working on a topic, you can do a search for the papers, and you can save that search. And when you save with this option, it will essentially search, I don't know, every week or so. On when there is a new paper that matches your search, it will send you an email. So you get informed when everybody is working on the subject. You don't need to do the search every week. The computer can do it for you. So if something happens, you will know.
Like a
Automatically.
subscription?
Exactly. It is a subscription. So without paying, of course. Okay, Denise delivered, and I think nobody else. So, okay. Okay, we have the time anyway, so we know who delivered after we start speaking. So we are starting the class. So this is what I did in the meantime. What I did, I wrote things here in this. Well, you know, this is advanced search, no? And instead of looking search, I put add to history. That was a quick way to tell me what was the number of things. So I searched for down affiliation. I didn't check the detail, but here they're probably a mix of down university and down street. I don't have a query to separate them here. Location didn't work, as you see, got 0. Then I looked for the doctor down. People don't write doctor, so the keyword doctor should not be there. What I did is to choose here author last name. Author last, since we had that option. And I got 150 Brown as author, down as author. Then when you search for Down syndrome, let's say if in all fields, if you don't say anything and you say Down syndrome, I essentially have 38,000 things, almost 39,000. But that is not necessarily connected to things. And you see the difference. If I look in the title, I have 9,000. And if I look in title and abstract, I have 14,000. And if we look like mesh term, you remember what was mesh term? Mesh term is organized a kind of more professional way to look for something, in the sense that every paper get classified by a professional, like a librarian, that put an official name that what is the topic of this paper. Because people can write Down syndrome in different ways, or can write, or maybe yeah, Down syndrome has other name. So it's not Down regulation, for example, and you have different topic inside Down syndrome. Or maybe because you have sometimes the rest that you are speaking about, the paper speak about one disease that is similar to Down syndrome or connected to Down syndrome, but it's not about Down syndrome. So the point that a paper says Down syndrome somewhere in the text, it doesn't mean that the paper is about Down syndrome. So you see, these 4 search give you different papers. The one that have in the paper, in the title, in the abstract. Here you have papers that speak about Down syndrome, even if they don't say Down syndrome neither in the paper, neither in the abstract. These are papers that say Down syndrome somewhere, but maybe they are not exactly about Down syndrome. So basic point here is learn to use these fields. And here you have a link to the help. So I essentially went to Pubmed and CVI, and then I search advanced, and then you have the help. And the other point is this. Remember, first here you choose the field. And when you get, let's say, journal, once you choose the field, you can see the options. What are these things? So say, let's say, infectious. And we'll search everything that are connected, every valid words that you connect with this journal. This is the short name of the journal. Infectious Disease Clinical North American, Infectious Disease Clinical Practical Baltimore, Maryland, whatever. So choose the field here, test the index. And the second point is you don't need to look directly. You can add to history and see how they are going. And then when you find something that makes sense, then you can click and see the exactly one that you want. So here you see all the Dr. John, Dr. Down, and you can see how they publish. There is a Dr. Down, very old. So, of course, since the last year when the papers are published electronically, it is much easier to put in the database. But some important historical papers are also published here in the I mean, they translated, or at least they index them, they have them in paper, but in the database, you can have them indexed. Okay, what else I see in this? There was something when I search help. Well, I don't have it at hand, but there is an explanation. The difference that Pubmed index everything, but doesn't include text, and PMC is only the text that can be shown free. So sometimes some journals give you the paper for free, but they are not in PMC. So PMC not necessarily has all the papers, all the free papers. What we have here, free article. Well, anyway, So my point here is that before looking at your search, you can see the numbers here and see if this number makes sense. Here it says 0 doesn't make sense, so we know that doesn't work. Affiliation, yeah, maybe take a look here. I don't know if it will be shown here. No, it's not so easy to see. We need to change the display option, no? Format, abstract maybe, I don't know. Here you see the affiliation. Porton down sulfur, yeah, there's the down. Darwinism. Yeah, it will be hard to find. In fact, affiliation, as you see, can be down or can be in, I mean, the institution or can be the place. It's hard to tell with this database. Okay. Any question before we start the class, the real class?
So
Any
about
camera?
the Down syndrome, was the mesh term correct answer?
What I did in my case, what I did was, I mean, what do you mean correct? Yeah, I will. If I was really looking for Down syndrome, I will go with mesh term.
Okay.
Kind of the control the vocabulary, no? That the one that will. I mean, it's very hard that a paper that has a mesh term is bad classified because someone checked it. Someone say, okay, this is Down syndrome, even if they use a different name, even if they write in a different language. Remember that people publish in different languages, so you maybe not say it in English, but you are looking at English, so the mesh term will try to correct that thing.
Okay,
and
all right.
people can write with orthographic error. The other thing that maybe we could show here, okay, it's not what I want. Let's go to NCBI, and we were thinking the protein. I was thinking the protein. No, I was thinking about. Okay, yeah. Okay, this is interesting because it's the protein that I want to use for the next of the class. So yeah, I said 10 to 50 sequence length. Many people did that, the same approach. And it says thousands, millions. So I clicked for the exercise of today. I clicked in PDB essentially because it was small. I didn't have any stronger criteria that having a small database that makes sense. And I found 25,000, 26,000, that kind of makes sense. It's something that you can handle. I just remember a slide that I had to show you. Well, I will show it in a minute. And well, this is the database, and I send it to file. You can do the same, or you can download from my webpage that I will give you a link in a minute. Okay. Now, if you wanted to restrict, that is here I got the result here. That you probably got the same way. The result, the real query is here, and that combines what I wrote in the header and the selection that I did in the site. No, you can also choose sequence length here and molecular weight, etc. Now, there was something else that I was thinking about. There was one question about the complete database. I mean, all the sequence between 10 and 50 that were millions, and then adding this filter that being in the protein data bank. Okay, then gamma protobacteria. So I'm not completely sure if this will work, but the way I will do gamma protobacteria is this. I will go to taxonomy, and I will say here gamma protobacteria. And in fact, I could have gone to nucleotide here. And Okay, let's see what's up. The problem is this. When you have a taxonomical thing in gamma protobacteria, you will have only sequences that are associated with the particular taxa. Normally, you want to see that taxa and all the taxa descending from that. So there is an option here like organism expand. Maybe I can show you in a different page. This is essentially everything that has the tax ID of gamma protobacteria and everything below. Okay, and here I will put something like 16s. Let's see advanced here. So organism expanded, and then we say gene name 16s 16s, and I will say auto history. Did I combine them? Probably had to combine them. Okay, why you think that I got so little 16s?
Maybe we should another column, not gene name.
Maybe. What can be? I think something else. The 16s has other names.
I used the title here.
Okay, not bad idea. Let's try title. 16s and organism. Let's say tax ID 1236 that we know is gamma protobacteria. Yeah, I give you more. But to be honest, I would like to use something like mesh term here because one of the problems is that the same gene can have different name. In particular, the 16S is usually called small subunit, SSU, or rRNA or something like that. That is usually a problem, that the gene names depend on the person who put the name. There's not a systematical way. So, well, probably for looking for the 16S, you will need a different strategy. Gene name. And the strategy that I want to suggest is the one that I want to show. Let's see what happened here. 16S title. Okay, that makes sense. Partial 16S gene. Yeah, kind of makes sense. So partial 16S. Remember that 16S tend to be partial. Why do you think that 16S are partial in this sequence, in this database? Any idea? Why we have partial 16S and not full 16S? Where are you?
They be unpublished yet? Maybe. I'm not sure.
Okay. It's a more technical problem. It's a technical problem that the 16S, typically the way that people sequence these ribosomal genes is they amplify with PCR. And therefore, you cut from the starting, the primers for 16S, they bind in very particular places that are super conserved. So if you are not using if you are using the universal primers, you will get something that is inside the 16S and you will lose the beginning and the end. So in most of the cases, you get part of the 16S. Why am I the only one with camera?
How do other people, I mean,
how people get the full? You can get later when people sequence the full genome, or in some cases people make an effort. But I mean, the easy way is just to get the typical amplification. I don't know if you have seen it, and we will probably talk about this later. But yeah, we will talk this in more detail later. But essentially, you have 2 or 3 regions in the 16S gene that are super conserved, and therefore people design primers there in the ultra conserved regions. Now the truth is that they are not so conserved. They are conserved like 99% of the thing, but if you have one bacteria that has not the same conserved region, you will not see it. So it's a kind of chicken and egg problem.
I tried this search with all filters as on 16S, and
okay.
I got more results, like complete.
Okay. Maybe the question is looking in here if we have RNA. Okay, let's say what was. What, do you want to share your screen, or do you want to tell me what was your search?
I tried 16S as a all files.
All fields.
Column,
Okay.
yes, and organism as gamma Protobacteria.
Yeah, here.
And I searched like that.
And we'll get more things. I agree with that. Now, sometimes the problem is that if you search in all fields, you don't know if the gene that you get is really a 16S or something that, for example, you may get a segment of several genes that include the 16S. Or you might get all ribosomal gene except the 16S. So if the word 16S is in the text, it doesn't mean that the sequence corresponds to 16S. You have to be careful with that. You have to be careful that sometimes the name appears somewhere in the text, but it's not always. So you need a second kind of quality control filter after.
Yes, I know, but maybe the difference is related with that because now in the results there are some complete sequences, but not in the other one.
Well, one good sign is this. You see the size here, 1000. The 16S gene, you know what is the size? It's around, the complete gene is around 1600. Like the 16S is like 1600s. The 5S is like 500, and the 23S is like 2300. So it's around that. So you know that it's less than 20,000. So maybe you can also put the range of, let's say 100, less than 100, we probably don't care. And let's say 2000 size of the search, no? Size of the, what is the name?
Sequence?
Yeah, size of the sequence, exactly. That will be a way to guarantee that we are seeing kind of only the gene, maybe complete, maybe partial, because it can be smaller. Okay. We will talk about this later because it's kind of super important. I mean, it's a typical thing when you get an infection and people do PCR to see if you got some pseudomonas or clostridium or something that people do most time is PCR of the 16S, no? Okay, we will talk about this later. So I'm going to start the presentation that I have for today, but be prepared because I will ask you a lot of questions. Fine? Yes, no?
Yes.
It's fine.
Okay. So I am opening this thing here. I am closing
Okay.
some windows. Okay. Okay. I hope my computer doesn't die with this. So full page. Okay. Search and comparing sequences. Okay. Let's tell the story this way. When you write a text, for example, in Word, let's see that you are using Word. You typically get that some words get with a red underline. Have you seen that? No. Okay. Yeah. People move the head. Good. What are these words? There's a word that are wrong, that are not found in the dictionary. Typically, there's an option add to the dictionary. So even sometimes the computer can tell you, you want to say this. You are missing one letter. You should say this one. No? Have you seen that? The kind of an automatic suggestion. How that works? How can the computer do that? Question. What do you think? So how the computer know that you are writing a correct word, and how can they tell you what is the correct one that you should have said instead?
Is it comparing with its dictionary? Like
Okay.
book is in its dictionary, and I write like book with 3 O, and it said this is close to this.
Okay.
It's
Very
comparing
good.
letters maybe with the ones and zeros.
The ones and zeros, let's say the ones and zeros are going to represent. We say that in the computer every letter is with a number, but at this level we can think about letters. Okay, they are ones and zero at some level, but if you look from some distance, they are just letters, and you compare letters to letters. And yeah, I like that answer. Anybody have a different opinion? Do you agree that the computer is looking at the dictionary and looking for a similar one? No, what do you say? If you say book, they will say that this. So, in general, there are two approaches, like philosophically. One is to make a program that kind of, sometimes you have a rule that defines a word, like, for example, you can say that in English, you never have 3 letters together. You can have the E repeated 2 times, the O repeated 2 times, but you have the A repeated 2 times is probably not a correct word. You can have a kind of automatic rule that here I represent with a function that takes a word and give you true or false. In general, that is hard because human language, or biological systems are complex and they don't follow easy rules. We will try that approach later, but there is a second approach that you can have a database or a dictionary, as you say. And that dictionary has the word that we consider to be correct, the word that we know. The basic problem is that you need to have that database before. So someone has to tell you these words are correct. It has to say what is the correct English word, what are the correct Turkish word, whatever. If you don't have a dictionary, you cannot. In fact, it reminds me many years ago, before your parents meet each other, I learned to use my first text editor, and they had a spell correction, but they didn't have a dictionary. So I got my text and everything was wrong, but then I checked it one by one and I was making my own dictionary. Like, okay, this is good. And I say, add to the dictionary. So for a while I was adding to the dictionary and I make my own dictionary, and I don't remember, but let's say 5,000 words. And the truth is, the language has much more words than we can talk. When we talk, we use a very limited vocabulary, and the language has much more. So, how many words do you think you have in English or in Turkish?
In English, I think it's more than 600,000, as I know.
I have a dictionary that I use in class, and it has in the order of 30,000 words. But there is some trick, like, for example, verbs, they tend to follow a rule, especially regular verbs. So you don't need to say all the variations. In Turkish, you also kind of we have a lot of words in Turkish, but they are following the same rules. So you can combine them in very well-defined ways. So it depends a little how, if you count everything in the word or just the key parts. I mean the stems. Okay, my point is that search sequence is essentially the same problem. I don't know if you see it immediately, but essentially we need a database and we need a way to search into the database and find if the sequence is quotations wrong. If it's not 100%, we want to find what is the closest one. Okay, so let me give you a couple of definitions because we are going to use it. When you search, the text that you are searching is called query. In this context, in the context of searching into sequence databases, query is the sequence that you are looking. Okay, query in general is the thing that you are looking for. In the first part, in the Pubmed, you have all these brackets. That was the query. Here, query means essentially the sequence that we are looking for. Then database, well, is the set that we are looking for. Right now, database will be just a FASTA file, okay? And when you go to the database, is a set of sequences, and each sequence that we take is called subject, okay? So when we compare a query and a subject. Subject is understood that it's a sequence that came from the database, okay? Just to define. Okay, so I prepared this. In fact, I told you how I did this. No, I went to NCBI. I say I want proteins that are less than 50 nucleotides, and then I say that they want them to be in the protein data bank. So we know that they are not too many, and I ask you to please don't load them. I hope I didn't test the link.
It can't be related with the address, maybe. Isn't there a hyphen, I guess, between dry and lab?
It is clever girl. Clever girl. Yeah, you are right. You see, yeah, but that is the real question that I put the wrong address. Okay, all my fault.
But right now, when I put
Let's
the
try.
hype, it says page not found.
Yes.
That is a better thing. That is a better thing. I mean that
Doesn't exist anymore, it says.
Okay, but at least this is the correct website. Yeah. Okay.
Can you upload to the blog?
I upload. Maybe I could have put in Google. I'm sorry, that is something I did and I didn't upload to the correct place. Okay, I think they will be available in a few minutes because it takes some time to update. But in the meantime, let's do this.
Another idea, we can share a file in the chat. You can send us through chat.
Okay, better idea. I like it, your idea.
Thank you.
Good. Well, thank you. Come on. This is for teamwork. Remember, teamwork. This is teamwork. Okay. Good. I'm sending a file. Let's see if I can send you the file, and let's see if it's not too big. Okay. Good. Did you got the files?
Yes.
Yes.
Good. Okay, I don't know why they are not available here, but they will come eventually. Okay, let's fix this later. Can you see this file in your computer?
First one, yes, but not the second.
And how did you show? Can you show? Can you share your screen? I stop sharing 1 second.
Can you see it?
Let me see. Yes. Okay, you got the CSV and you open with. Okay, interesting. But there is something that you may notice, that everything is in one column. It didn't split. When you import, you have an option. I will try it, but that is the kind of thing that I wanted you to see. Good. I like your result. It is still need to be improved, but this is the idea. Who else got? Did anybody get the same, or hopefully something different? And if not the different, at least the same.
Mine is the same with Oslo, but the second one is not opening also.
Me 3.
Okay. When you say open, what you do to say that you open something?
In Turkish? What do you mean?
No, in the computer. What do you
Open
call
file.
opening? Open. Okay, open file. Let's say.
Can
It
we
was
open it
shown.
with Artemis? Maybe. Can we try it?
You can, but it will not work because Artemis essentially take one sequence and you have kind of many sequences. Where are we?
Do you mean open as, like we can open it with another application or something?
Exactly. So, okay, I'm going to share my screen. Do you remember that I say that we need to use a text editor, and I recommended you Apple Atom or so some way. I say two things. We need Artemis and we need a way to see text files. So this is my editor, this Atom. I also recommend Visual Studio Code. And a text editor, as we say before, you can even open with the R editor, at least the small file. I don't know if this big one, but the small file you can. So this is literally what I got from the NCBI. Okay, I went to the protein data bank and I downloaded this set of sequence. What was the name? You remember I say between 10 and 50 nucleotides and in protein data bank, and give me the FASA, and this is what I got. So if you have to do it again, we can go to NCBI and repeat the query. And I got this. And I load in R and I process it a little bit, and then export it, and I get this comma separated value here. So the first thing that you should always use is some kind of tool to see text. Remember that most of the time we work with text files, and you need a way to see the text, what is happening inside. If you get a FASTA, GenBank, or many other things, you will need to have a look what is happening. And if it's binary, well, at least you will see that it's binary and you will know what to do. So this is a starting point, okay?
Teacher, my Artemis, I downloaded it and I unzipped it and I downloaded the thing that Ahmed said last week, but it's not opening anyway.
Okay, we will try that later. We are not using it yet, so I'm giving people try the chance to see it. What we can do, and same, what I don't know if Irem is here today, is we can agree an extra meeting after the class that you share your screen and we see what happened. But
I could open Artemis.
Good. So, but in this case, Artemis is not helping us because this is a FASTA of protein, and Artemis works with FASTA nucleotide. So it's not the correct tool for this.
Okay.
Tamam? Fine. So let's see. So my point is this: when you open a file, when you use window, people think that opening a file is double click on it, and it will do automatically, because in many cases, in Word, in Windows, the files that are associated with the application. But in general, here we will have files that can be opened in different ways. And you should be able to kind of open the program first, like Atom or Artemis, and then from the program, open the file. So instead of double clicking the file, try to double click the program, and then in the program, tell what you want to do. Okay, that is a first change of approach.
Artemis is just that way, right?
Yes.
You can't open directly in Artemis. You need to first.
I think we will not even open in Artemis because Artemis doesn't process this protein sequence.
Yeah, I know. I just say.
So in theory, in Windows, essentially Windows has a table that says if the file ends with, I don't know, doc, then you open Word. If it ends with Excel, you open Excel. If it ends with PPT, you open with PowerPoint. If it ends in PDF, you open Acrobat Reader or something. There is an association. But that is something kind of to make life easier or complicated, depending. But it's kind of to make it automatic. What I am saying that you should be also able to open the program that you want to open, and then from the program that you want to open, you go to kind of File, Open, and you open the file that you want, and you can see in the context that you want. So maybe I will close this one now. Just to, let's see. I mean, if I'm not running out of memory, I'm not going to. But an alternative was to open with R, for example. So if you open here, you have file open. You probably can go and open the sequence that I say here. What is this? I have it here in static. Bioinfo. Short protein CSV or short protein. Let's see if I can open the fast amino acid here. It says big, but okay, let's go ahead. It will take a minute because RStudio has a text editor, but it's not very efficient for big files. So Visual Studio Code and Atom are much better for big files. Anyway, let's go to the real thing. The point is you have the sequence. And okay, no, there's something else I want to show you. Let me close RStudio for a second. We'll open again if necessary. And the Excel way. So I will open Excel for a second, but in general, I prefer to use the one that's online. The Google thing. Okay, and then after you finish, I want to search some text. Okay, this is the thing that we're going to do the search. Okay, Excel. Finally, here, let me open. It takes so much time. So let's see. Excel. I am copying to the screen. So you see my Excel, no? So here I will go. If you want to do the opening in Excel, open, because the CSV is made to be open with Excel or something like that. CSV means comma-separated files and comma-separated values, and it's a way to put what is the name? It's a way to put data in a text file that can be easily read by a thing like Excel. So yeah, it's sometimes, in many cases, kind of what we want to do, kind of organize the information in a good way. So you see, CSV is one of the formats recognized here. So let me open and okay, look, format. Okay, don't care about this. If you do it correctly, you should get 2 columns. Why 2 columns? Because there are 2 parts that we care about. One is the, let's close this thing. One is the sequence, and one is the, how can I make it smaller here, this thing? here. Okay. One is the sequence and the other is the name of the sequence. And in general, we want to keep them separated. Okay? Because one is the sequence that we are looking for and the other is the name. So in general, for example, if you are looking for this text that I show you in the query here, if you want to look for this text, what will you do? How you search for this in Excel?
I use Control F. That's kind of cheating.
Exactly. Control F, and you will give some text there, and you say search, and you will get one that is exactly the same, no? And then what is this? It is contained inside. Interesting. And you have an advanced search that we will look at. Probably in advanced search here we have the option like find the complete cell that match only the full, not sub part. You see the difference. If we say find entire cell, it looks exactly the same text. If I don't put that, I can also find another sequence that contains the query, okay, like a substring. Okay.
Does quotation marks work here?
Yeah. Quotation mark in which sense? Sorry, I cannot hear you. You're muted.
To get rid of the other sequence, the second one that contains this part.
I don't think that the quotation mark will work in that sense here. Because a kind of quotation mark is part of the text that we are looking for now. Okay, there is a comment, but I think I will give it later because I don't want to get. There is a point that when you imported, since I separated by comma, and I'm using, I use my Excel in American format, that comma, I mean, that I use dot to separate decimals. And in Turkish, normally it's not the case. So there is an option, but I will show you later. I even have a video about that. So let me close this Excel. But these are the two ways that you probably can do it, or three ways. We can open with a text editor and you see it directly. And the search editor also has a search. You can open it in R. In fact, it's not very hard to open in R. Both cases are easy to open in R. And then search inside the R. Okay, anyway, so you should be able to do this search. And I will tell you the point. If you search for this, as we tested, you find something. If you search for this one that has a one letter change, you don't find it. And you don't find the one with a letter inserted or with a letter deleted. So the first query gives you one answer, and the second gives you 0. Okay? Agree? So, well, that was what did you find?
Can we do the search on atom?
Of what?
On atom.
I don't understand you.
Atom.
Atom. Atom.
Yes.
Okay. Yeah, you probably saying correctly. Yeah, I think that you Control F probably, and you say the
okay.
What was?
Okay, thank you.
And I put replace the Y by A, and I get not or YA, and it say not found. But yeah, valid question. You can also search here. Okay. The point is this, going back to the presentation, you know that this sequence is the first matched, the second didn't match, even if they are very similar. So you know that they are similar, but we didn't find it. So how can we find sequences that are similar but not identical? That was the question. Like you wrote a word that is not correct and you want first. So at this level, we're essentially saying, okay, the first word is correct and the second and third are wrong because they are not in the dictionary. But then you want the second part. You want to say, okay, if this is not correct, what is the correct one? What is the closest thing in the database that matches the query? Okay, agree? Okay, then I have another question for you. What do we mean by similar? When are two sequences similar? By the way, I am missing the rest of the people here. I should have asked I should have made the quiz at the end so they don't run away. What do you mean by similar? When you say that two sequences are similar, when you write book instead of book. Any suggestion that what we call similar?
Well, it could be dependent to the size, but let's say if 50% of the sequence is the same, it's maybe.
Okay, how you count 50% of the sequence?
What do you mean?
Yeah, what do you mean 50% of the sequence? What do you mean 50% of the sequence?
Let's say if we have a sequence of 100% is half of that, so.
But what do you mean being equal?
Like the nucleotides, maybe?
Okay, nucleotide or letters. Okay, but let's
Same
be more
repeats,
explicit.
maybe.
Okay, repeats is a little, you are introducing a new concept. At least nucleotides or letters are simple. Repeat, it can be, but you have to define what is a repeat. You get into complicated waters if you go there. The question becomes more complicated. Okay, let me. I mean, Ahmed it's right in the spirit, but then you need more definitions because you are making an assumption that you are not declaring. You are saying that I can compare things, and they say that I can assign a percentage of how similar they are, but you don't say how you calculate that sim. You are saying, okay, if they are 50% similar, they are similar. Okay, but what is 50% similar? 50% of the well. Okay, so let me give you the classical answer. We can calculate a distance between sequences, between strings. So the first idea is that you take two sequences that have the same length, as you say, and you count essentially how many times they are equal, how many times they are different. Okay, you define this to be a distance. So if you are very close, it is good. If you are very distant, you are more different. So the closer you are, the similar you are, the more separated you are, the more different you are. Okay? Follow? Yeah.
We are not talking about a physical distance, right? Just
well, depend of what you call physical distance. These are letters in the computer, so they are physically nowhere. We are not speaking about Euclidean distance. We are speaking about distance between. So, for example, you can say distance in time. For example, we say that Cleopatra is more distant to the pyramids than to us. For example, you know that that Cleopatra is more closer to us in time than to pyramids. That is. So you can measure distance in you of course you have the physical distance, but you can kind of like, I don't know, in music, you meet someone and you find that you have the same music taste, and they say, I am very close to this person in the music sense. Or you say, I am very close to my family. I have my close friends. We use the concept of distance in an abstract sense beyond the three-dimensional thing. And that is a super important point. That is going to be one of the topics that I will speak a lot, the distance between things. I'm not giving you the complete definition, but it's going to be important. Right now, the point at distance for now is something that takes two things and give you a number, a positive number, or not negative number. Okay. So in this case, it's very easy. The distance is how many letters are different. Agree? So you can count the number of mismatch or the number of substitution, if you like, the number of mutation, if you like. Biologically, they tend to be the number of mutation. And if distance is 0, it's perfect. This has a name. This is called Hamming distance. Hamming is the person who defined this in the 19th century. I don't know when, but it's an old mathematician that defined it. This is Hamming distance. Basic thing. Essentially, you can say it like the number of change, the number of letters that are different, but you can also say the number of steps that I need to do to go from one to the other. For example, how I go from cat to cat, I am immediately distance is 0 because they are the same. Cat to bat, you need to change one letter. Cat to back, you need to change 2 letters. So the Hamming distance can be seen something static or something like an operation. How many things I have to do to go from one place to another. How many steps I have to traverse. Okay, that is a way we measure distance. What is the distance between your home and the university? Well, how many steps you have to take? That is a way to measure, no? And you of course realize that if the distance is 0, it means that this is a perfect match and the word is correct. And if the match is not 0, how you it means that you are close enough, and you will try to find. If you don't find a perfect match, what is the closest one? That is the way to solve the problem. Okay, you see the point? Agree? Yes. Yes. Okay.
Yes.
Let's make how you make this in Excel or in Google Sheets. How you will make it?
We can try advanced search and Control F command.
No. In Excel, in the cells. No. Control F. I am doing in a new page. I'm thinking about this. Let me escape here. How I get out? Here. I say no. Okay. Exit full screen. Yeah, exit full screen. Yes. So I'm opening a new tab and I say docs google.com sheets. Okay, sheets. By the way, I am not a native speaking, but it is important that this sheet be with a long E, otherwise you are in big problem. You are laughing with Sanya, but real story. It happened to my aunt. My uncle went to study the PhD in US, and she went to buy sheets, and they looked funny at her. Okay, I suggest that you do something like this because it is important that you practice with your fingers. So this is okay. Let's say class 5 bioinfo 2020. Okay. Eventually, we can import the sequence here, but my idea is this. For example, I have this example here. You cannot see it now, but let's make this mouse and grows. This is not Hamming distance 6. This is wrong. Hamming distance 1. Okay, but let's do it. So I will put M-O, 1 letter by column, and then the other was grows. Let me move them here. So. Oh, no, this is fine. Yeah, I'm right. No, this is correct. Okay. There. Yeah, it is true. In fact, let's start before this. Let's start with the one that I give you before. Just 1 second. Let's start with the one that we have said. Cat and bat. Okay, let's start with the simple one. Okay. Agree? And then we go to the this is a little more complicated. Agree? Let me make the letters a little bigger. Good. You realize that I am looking at the screen and not at you, so I don't know. So I say cat, C, A, T, 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 and bat. Okay. So how you do? What I will do here is to compare this, compare the first and ask if it's equal to the second. No, if it's different to the second. Not equal to the second. Not how you say not equal, you say different. You say less. Yeah. So are they different? Yes. Okay. You follow what I did?
Can you show it again? What did you
Yes.
write to say not equal?
I put less than
Okay.
and greater than. That is the Excel way of writing different. It's kind of, because Excel is made by Microsoft and Microsoft make it in BASIC, and that was the basic language structure. So it's either bigger or smaller, but not the same. And then we can, if I am not wrong, we can sum, and true will be 1 is to, no, didn't work. No, so it's not what I want here. So I need something a little more different. I want to say if A1 is different from A2, I will give a 1, otherwise I will give 0. Okay. 1 and 0. Okay. So let's say that each column here is giving you a point that can be, and this is going to be the idea. This is an important idea I want to show you, because we're going to do this much more, but this is going to be the basic idea. You have the two letters, one in front of the other, and you are going to get a number representing if this is a good or bad. So if you have cat and cat, they are all equal. Every column is giving you 0, and this is the total. Let me move it a little bit here. Maybe I will put it here, and I will call it this Hairpin distance. Okay? And we can have longer letters. But let me show you the formula again. It says, if the first row is different from the second row, then give me a 1, otherwise give me a 0. Okay?
Okay.
Why does it say error in mine? I write the exactly same thing.
Maybe you need to put semicolon, depending on your language. Since I'm using the American convention, I can separate with comma. In the you may need to put like this with semicolon.
Okay, I will try different versions.
Try with semicolon. Try with semicolon. And if you put then but, you get 1. If you say bag, you get 2. So it's a way to measure. Okay, then I want to go to the next example that I was talking about. Yes, no?
Yes.
Okay, so going back to, so I want to take this to mouse with, okay, but I didn't read the title, but you read it, no? What happens if the sequence have different length? Because in this case, to make this comparison, the two sequences have to have the same length. But in real life, some words are shorter than others, some words are longer, some proteins are short, some proteins are long, some genes are short, some the length is not going to be always the same. Okay. So what we do in that case, we insert an extra element that we call gap, and it typically represented by this dash. Okay. Dash will represent gap is something that we put to fill, a filling that we put to make the two things have the same length. Okay. Agree?
Yes.
So let's try this. Mouse with gap. So I say M O. Why is not working the big caps? Okay. M O U and no O. And here I need to put the here I have to be careful. I have to put quotation hyphen. No. Okay, work. Sometimes it can get confused. I need to copy this here, and I need to update this to count until F. Okay. Having distance 6 because kind of everything is different. Okay. Can you see how this can be better?
If we put this dash before the mouse, not after the mouse.
Exactly. I will copy this, paste it here, and put the dash here. And then we have distance 2. You see, no?
But isn't this basically against the rule because these two strings aren't the same length, don't have the same length?
Well, well, you yes and no, yes and no. In the sense that yes, it's against the rule, but is the point that we're extending the rules. In the Hamming distance, things have to have the same length. And since we want to do with things with different length, how we expand the idea? How we extend the idea of Hamming when things have different length. And the solution is that we have to put gaps. So yeah, we break the rules, but we break them to extend them. We are making new rules. So let's say that we are not rule breakers, we are rule makers. Okay, we are making rules. Okay, extending the old one.
Can you show how did we submit as Hamming distance in the Excel?
Yes. So there is two parts. One is below every column, I put this formula. That,
yeah, I made that.
Put it here. And then here I essentially say sum of all the elements in that row.
Okay, so we didn't define as Hamming distance.
No, that is I put a name Hamming distance, but the Hamming distance is the sum of the values that you get in every column, and the value in every column either 1 or 0, depending on the if the letters are the same or not. Okay.
Is there a way that we put the name to the calculation, or we should
that define a new formula? I mean, the short answer is no. The long answer is there is a extension of every language, and it's the kind of thing that people get in advanced courses. I mean, it's a course by itself, how to define new functions in Excel. You can, but it's not something that we do normally. It's not so easy. So. And the other point is that we are not going to do it very much. I'm showing this so you can understand how the things are doing. This is a little practice. We are not going to do this in real life. We are just using to understand the concept. Okay. Now, we tried this, and you see the Hamming distance is a gap. So another definition: put into sequence face to face is called alignment. Pairwise in this, because there are two of them. We are taking two sequences and we align them. So what we do here and put in the gaps is an alignment. So we have two different alignments. The original one, this one that has M with G. And let me put it in a more bold letter so we see it more easy. And kind of, I believe that if I make it more narrow, it will be easier. Maybe something like this. Maybe I can put them in the center. Yeah, something like that. Okay. Maybe I will put this number here. I can make it a little narrower. And then we can zoom here. What is zoom here? Fine. So here it was very bad, but then I did another alignment. In other words, I choose the gaps in a different way and get a better having distance. So, okay, let's go back to the text. So there are many ways to insert the gaps, as you notice, and the distance will depend on how you put the gaps. You realize that we solved the problem. I mean, the problem that we have is that we can only do having distance when things have the same length. Since we want to compare things of different length, we were forced to do something. The thing we do is insert gaps. And the problem that we get is that we have many ways to do the gaps. We replaced the problem of distance to the problem of where to put the gaps. And you can put in many ways and you will get different distances, different values of having distance depending on where you put them. Okay?
Okay.
Understood? Yeah? No? Okay? Okay.
Yes.
Okay. If you don't have camera, you have to speak. If you have camera, you move the head. Move the head. Okay. There are other 5 people that are here, not camera, not speaking. So let's see what will happen with them. So
this 2 words is kind of short, and we can see the similarity here. But what about bigger sequences? How we determine where to put the gap?
Yeah, it's going to be in this case you can kind of do by hand, but in real life doing by hand is impossible. So let me give you a difference, and I prepared a video for this. Why it didn't work? Oh my gosh, why things don't work? So when you have a line here and you have a dot, you can have the distance between the dot and the line. If you remember correctly, you can essentially make any path between the dot and the line, but the one that we call the shortest distance, the one that we call the distance is the length of the shortest one. So there is one that is the shortest, that's the one that's going yabash. So okay, that is the bottom line here. Let's I should go back. So this one. The distance between a point and a line is the shortest length from what is this thing moving? Stop. Yeah, I could not make YouTube to not show the publicity. The distance between a point X and a line is the shortest length from. So if there are many ways to traverse, if there are many ways, let's say from going from your place to the university, let's say, the one that we call the distance is the shortest one. Because you can take the shortest, or you can take a detour and you go to somewhere else and you take a lot of time. So the shortest one is the one that we call distance. So we are going to use the same idea. If there are many candidate distance, like many having distance, depending on how we put the gaps, we choose the smallest one. The distance that we're going to say is the one that we put the gaps in the best way, the optimal way. So again, the distance is the idea. Well, this is the video I should show. If you have 2 points, A and B, I'll make the video faster next time. You can have many paths between A and B. They're not necessarily the best. So this is equivalent to putting gaps in the wrong way. Okay, if you put the gaps in the wrong way, you get distance that are longer, essentially because you took the detour and you didn't went to the good way. The one that we are going to call distance is the short one. That is the straight line that you see here. Fine. So the point is that we are, since we are adding gaps, and we define the distance in this way. Our definition of distance is we put gaps and we find the smallest combination. We find the combination of gaps that will give you the smallest value. So this is not longer a Hamming distance, and this is called, do you know the name of this? This is called Levenstein Distance, which is another person who invented this thing. So just to give you an idea, here you have two words of the same length. If you use Hamming distance, you have 10. If you use Levenstein distance, you are allowed to put gaps, and then the Levenstein distance is 2. Okay? Turkan?
Okay.
Turkan has a question mark in the face.
The first one, okay, all letters are different and the Hamming distance is 10. That's fine. But I didn't get the second one because
in the second we're allowed to put gaps freely. And in fact, more than freely, we have to put gaps in the best way possible. So we put gaps trying to reduce the number of mismatch. Okay.
But the Levenstein distance says just 2 letters are different.
Yeah, we have this A with the gap and this gap with the A. Okay. Biologically, this means that let's say, well, we say insertion deletion, but kind of we don't know what happened first. So from the first point of view, this is a deletion. The A got deleted, and this is an insertion. There was nothing here, and we inserted an A. Okay, so the gaps in the query are normally insertion, and the gaps in the subject are normally deletion. But since we don't know what happened first, we normally call them indels or just gaps. Okay, but they have a biological meaning.
Is there a way that we compare two things with different lengths?
Yeah, put in the gaps. Put in the gaps.
But if we didn't put the gaps, does it make the comparison?
No. Well, yeah, you can invent things, but we want one that represents something meaningful in biology. We put gaps because the gap have a biological meaning, have the meaning of insertion and deletion that that can happen through replication. So yeah, in theory you can do that, but we want to choose a model that represents biology. So
can I ask something?
Of course, you asked it already. So
what if we have too many insertions, like it changed the sequence length too much?
What do you call too much?
To the point of we can't see the sequences actually similar, but there are too many insertions, so the first sequence maybe is too long.
I mean, the distance works always. It's like you are essentially asking, what is the distance between two cities is too big? The distance between two places is too big. What do you mean that being too big? Distances are distances. They are never too much. Unless you have another criteria for that. But the distance is just measuring how far away are two things. And there is no concept of what is too much different. It's just more different or less different. Maybe you have another concept later that say, okay, if the distance is too big, then, for example, you say these are different species or different proteins or something. There is a moment that you say, okay, when a word becomes a different word. because in some sense, maybe the point is this: everything is similar to everything in some distance. We are all close together. In the same way that we can say that since we all descend from the same ancestor, we are all family. We are all related somehow, but at different distance. I am very close to my brother. I am less close to my cousin, and I'm much more distant to Donald Trump, but it's part of my family, no? So distance is something that you can always measure. When that distance is meaningful is a different question that will come later. You follow me? So, Basil?
Okay, I guess.
So, I mean, you can always, the point is that given two words, you can always measure the distance. And
you say
the
use
distance can be
gaps.
millions.
You said we can use gaps if the lengths are different.
Yep.
But how can we know if there are too many insertions or deletions and the length is too different?
There is no such thing as too many. There is no such thing. There are many, but never too many. Because you are assuming that there is a limit that there cannot be more. No, it can be 10,000 billion, trillion, 4 trillions. Just a big number. They are never too much. So, for example, you can have a gap, and then you have gap B, gap. Well, okay, maybe here you have a point. There is a rule that I didn't explicitly say, is that you should not have a gap in front of gap, because that doesn't make sense. If you put gap in front of gap, you can delete them, and they I mean, they are not biologically meaningful. They are not meaningful. Gap in front of gap is like you are forcing them to I mean, they don't have any meaning, no? You are forcing them, but they don't have any meaning. Yes, no?
But with her question, actually, she's got a point, because when it's about sequences, how will we know that where to put the gaps if there is really too many deletions?
Okay, let's go to that. Let's go to that. If it can happen, you can get to this point that you have gaps everywhere, and they are very different, but sooner or later you have one letter that is common, because the alphabet is limited. For example, in nucleotide you have 4 letters, so sooner or later you are going to run out of letters, and you are going to have an A in front of an A and a T in front of a T. So there will be always something in common. In protein, you have 20 letters.
But how will we find it? Because it's thousands of lines with the sequences.
Okay, very good question. Okay, in fact, these are the 2 slides that I have now. First is like what you are saying. We can put gaps in many ways. The way that you can put gaps is very, very extensive. And I'm not even counting. I am saying that if you have m letters, you have 2 to the power of m plus 1, because you can. If you have 3 letters, you have 4 places where you can put gaps. So you have between 0 gaps and all gaps, you have many combinations. But then you can put 1 or more gaps, so you get something like this. Yeah. So how to make it in a smart way? Okay, we're going to see all the details later, but the first idea is this. I'm going to show you. Instead of putting them face to face, we make a rectangle, the query in the rows and the subject in the columns, and you mark the cells that are equal or different. Like here I put A, B, C, D, E, F, G, H, I, J. And here I have B, C, D, E, F, G, H, I, J, A. Okay? So here I am comparing an A in the first position with the A in the last position. B in the second position with B in the first position. Okay? Let's took a minute. This is the kind of the central idea of today. You have one word in the rows. Every column here has a letter. This is representing the letter A. This is representing B, C, D, until J. Okay? Same the rows. The row represents the letter B, C, D. The one that you can read them here, no? And when the letter of the row and the letter of the column are the same, then I put a black dot. Okay?
Okay.
You see how we do this, no? And we can try to do. Then you know that the black dots here mean that the two things are equal. So if you can move from here in the diagonal, if you can move by the diagonal, you are fine. If you move from black to black in diagonal, then everything is free. Okay? But if you move from white to black, you pay a price. So here you pay 1 from white to black because you start from the corner. The goal is you want to start from this corner and you want to get to the opposite corner. Okay? So you start here and you have to jump. Then you can move free and then you have to jump again. One horizontal jump, one vertical jump. Okay? So you see that the horizontal jump is a gap in the second. The vertical jump is a gap in the first. It's this one. These are the letters. This alignment that I put here is the one that I'm showing in the graphic. Okay? So Excel. I will make a new one. And we can try something easy. So what I'm going to do, I'm going to start here and I'm going to say A, B, no, big letters. No. They don't need to be big, but you have to be coherent. A, B, C, D, E, F, G, H, I, K, J. Okay, let's make them narrower. And same here. B, C, D, E, F, G, H, I, J, K, A, no?
KJA.
Sorry, I'm bad. H, I, J, K. So this K should not be. In fact, I make a mistake here. Delete. Just to make it shorter. I mean, you can make it longer if you like. Okay? One in the columns, one in the rows. Okay?
1A isn't.
Because I forgot. A here. Yeah, I forgot. Thank you. You recognize that this is one of the texts that I show you in the slides, and the other is the other text, no? Let me go back to the slides. This one. I put K. Well, anyway, let's put K. Let's put K here. Let's put K and then A, just to be coherent. Tamam? Agree?
Yes.
And then what you fill inside? You do similar as before. If the row, if the column name is different from the row name, then you get a 1, otherwise you get 0. Fine? Okay, there is a trick here that if you are good at Excel, you will know. If I copy this, I will get in trouble because the letters change. This say, you see, this is taking instead of taking the row name, is taken to the side. Huh? You see, this is taking V2, but it should be A2. So before copying, you want to make this modification. You want in this B1, you want the A1 to be fixed because you want the row 1 to be fixed, and you put a $sign before. Same in the column, you want the A to be fixed. So you say B$1 and A$2. That is the most important thing that you have to know in Excel, that how you make a formula that get anchored. D, and the A is fixed and the 1 is fixed, and then I can copy down. Let me show you the formula again, just in case you miss it. Let me show you his one. No? If B$1 is different from $A2, so the dollar doesn't change the formula, but it changes how the formula adapts when you copy it.
So this is the opposite version of the two. This is the
Yeah, it is kind of transposed in the sense that
all ones should be
yeah. It is the typical problem that it happened because in when we do a plot, what's the plot here? When we do a plot, the x is going up, but when we draw a matrix, the x is going down. It's yeah. So yeah, so here this is B. This is growing up. This is the horizontal is fine. The vertical is in the opposite direction, but it's the same idea. Okay. Agree? Say something.
Maybe.
I mean, did you get the colors? I mean, the numbers at least. Did you get the formula? Did you copy the formula? Maybe I will copy the formula in the chat, okay? You may need to change the...
But we use Turkish, so it's not if or it's
yeah,
not.
semicolon. And the Turkish. Yeah, why you use Turkish? What do you think if I use Spanish here? I tend to, I used to use Spanish in my previous life, and people say, why are you using Spanish? Don't be silly. Yeah, they were right. No. Copy the formula. Why I didn't copy the formula? Yeah, one of the problem of Excel is that the name changing depending on your language, and that is kind of silly, but because it means that you cannot do it universally. But there is kind of some option, at least in Google Sheet, you can change your language. You know, there's an option that if you want to do it in Sanskrit or Malaysian or Yoruba, you can change it. There are like 3,000 languages in the world, maybe more. Do you want to use all of them? Worse. Do you know what is going to be the language of the 21st century? Chinese. Are you ready to speak Chinese?
What? How?
Because Chinese is the most fast-growing economy. They are very well organized. They invest a lot on science, and English. We speak English because British and Americans, and you see them. Okay, next trick here. There is an option called conditional format. You know, conditional formatting, which in this case I'm going to do this thing. If text contains 0, it will be blue. Okay, done. So I put colors now. There is something called, you have it in Excel. This is taken from Excel. It's called conditional formatting. The syntax is a little different, but you
Can we do this on RStudio with the plot?
Yes, sir. Yes, sir. Very good question. Very good question. Do I have a studio here? No, I don't have. But in RStudio, the plot that I show you here, as you know, you do everything in RStudio, or with R at least, in R Markdown. This is a function called .plot, and that is a function that came with the seq.in.r. You remember we used the seq.in.r to load sequence in R? That package include a function called .plot, and this is how I make this thing. So yeah, the short way, I didn't want to say, but the short way is to do it in R. That is going to be the easy way.
It's easier than Excel, I think.
I mean, to be fair, when you are just trying to understand, sometimes Excel is better. When you are exploring, when you are starting the problem, kind of Excel is good because you can try it a little. But if you are going, when you get the idea clear, R is faster to, not only faster to calculate, but also faster to express your ideas. Okay, my headphones are running out of battery, so I should be stopping. Okay, but let me show you. I will share this thing with everybody later. I can probably make it public now. Okay, let's do it now. I will change link to anyone with the link, and I copy the link down, and I am putting the link here, and you can see there. Okay, but try to do it in your own, because next class we are going to do it again. Okay, now finally, let's go back to the this is another sequence, and here you see that we start good, then we have to jump here, either two vertical and here, or we can go, yeah, this is the fastest way, kind of one here and then, yeah, but you need to cross by whites, and then
Teacher,
you have here.
with the link, we got the mouse graph sample. Is it true?
What do you mean?
With the link, it gives the mouse graph sample that we did in the first one.
Because you are open the first. There is a, in the lower part there is a button that say sheet 1, sheet 2. You see, they are different. Yes, no? Here, in the lower part.
Yeah, I got it, but it's not here. Is there anyone that can open the link?
Alt tarafta sheet 1, sheet 2 yazıyor ya Azal. Alt tarafta.
Tamam. Okay, thank you.
Okay, let me give you, this is essentially what I wanted, but let me show you the next, we don't have a lot of time and I am running out of battery, but the last point that I want to show you, you can make this plot in many cases, and the plot will tell you essentially how to jump. Maybe the point is that there may be more than one way to jump. The way may be more than one way to put gap that will give you the same distance. So there are two things here. One is what is the distance, and second, what is the gap. This is the mouse growth, for example. And here I have abracadabra. You sometimes want to compare the sequence with itself, that you do because you want to see this pattern. Okay, well, give me 1 second. Okay. Okay. Here I put abracadabra, which stay with abra and finish with abra. So this is a repeat that Arda say at the beginning. So Arda abracadabra. So how you find a repeat? You find a repeat because you have the diagonal. When you compare a sequence with itself, naturally the diagonal is going to be full because every letter is equal to the same letter. But if you have something outside the diagonal, I mean, what I mean, if you have diagonal points outside the main diagonal, it means that you have repeats. You see here? Yes, no?
Yes.
Okay.
But what if they're not symmetrical?
No, they are always symmetrical because if there is a repeat and since you are putting the same sequence twice, they will be repeated. Look like this.
Oh,
this is a more realistic.
Okay.
This is a more realistic. This is a protein that I found with the repeats. So I am comparing the sequence with itself. The expected result is that you are going to have this main diagonal fill, but you can see, and they have to be, since it's the same sequence, it will be reflected. So you have a repeat here and a repeat here. So you mean that this region and this region, you go, I don't have a way to draw here, but you go horizontally, you get to here, and vertically, and you find that this region here and this region here are repeated. And also here. So this is kind of an X-ray crystallography, if you like. If you put the same sequence in both sides and you see this pattern that shows you that there is some repeat. There is another important case that you also can find with this dot plot is something called hairpin. Do you know what is a hairpin?
Yes.
Or a palindromic sequence? Here I put a sequence and part of it is repeated backwards. No, they are typical. That is important because, for example, one way that transcription ends is when the RNA make this loop that is fold on itself, and it kind of detach the transcription factor, the polymerase. Yes. So it looks like a diagonal, but in the opposite direction. Not in the main direction, but in the opposite direction. Okay, that is also that you see in the dot plot. Fine?
Yes.
Finally, I want to show you a little extension of this. We will have another next class, but this is, by the way, the main part of genomics. This is comparing sequences, the most thing that we do. Levenstein distance is also known as global alignment because you essentially compare the complete sequence with the complete sequence, putting gap if necessary. Some cases, your query is smaller than the subject. For example, you are looking for a gene inside a genome. I give you this example. Here I have a long sequence, and here I have a short sequence that is partially contained here. So in this case, it will not make sense to go from one corner to the other, but what makes sense is going from one border to the other border. Okay? So, essentially, in the border, you can move freely without paying any cost. That will translate to this concept. There are two kinds of gaps. Internal gaps and external gaps. By the way, this is called semi-global alignment. It's not global because one of the sequence may be shorter, but the other has to be full. So in other words, we try to match all the short sequence, but only part of the long sequence. Okay? So internal gaps are gaps inside the sequence, and external gaps are outside the query. Maybe I should draw this. So external gaps are handled different here. What we say is here, if we move from the corner to the start of the query, all these gaps are going to be outside the query, are external gaps. Then you have a gap here, that is an internal gap, and then you have external gap here. So we distinguish internal from external gap, and the basic point that external gaps are technical issues. Typically, it's because you use a PCR primer or something, and they are part of the sequence that you don't see, but you don't see because of the technique that you use, not because of nature. It's because of technique. And the basic question that you want to solve in science, in particular in molecular biology, is separate the effect of the tools from nature. You want to say something about nature, not something about the tool. Therefore, in this case, everything outside the sequence doesn't count. External gaps are ignored. Internal gaps, on the other side, are made by nature, are replication or something like that. They have a biological meaning, so they must be counted. So that is the basic difference that we have in the semi-global. So this is the summary of today's class. We spoke about Hamming distance, that is essentially counting mismatch, Levenstein distance that allow you to put gaps, and you have to find the best way to put the gaps. You do that looking at the dot plot. And then we redefine a thing that Levenstein is also called a global alignment, and we extended to say local alignment, semi-global alignment. Semi-global is the one that can take external gaps for free. Okay? That is the summary of today's class.
Okay.
So next class we are going to extend this to more cases. Tell me.
Can you quickly show how to make a plot on this Excel to see from like your examples, see like your example?
Well, which one? Which example?
Let's say ABC, the one we did. Let's turn that into a plot quickly.
This one, for example? No.
No, the one in Excel. You have in Excel.
Ah, you want to do it in R? No, in Excel. How you want? I didn't get your question. Tell me your question again.
Can we make a plot of this numbers to see the black dots and the white dots?
But I put the colors directly here in the cells. I did with
it's upside down and it's not looking like a graph.
Okay, if you want to put it upside down, we can essentially the order that we put the letter here. You will say B, C, D, E, F, G, H, I, J, K, A. Sorry, A. Okay, now it's in the same order, no? It's just the way that I draw it.
Ah, okay.
And then the colors, you can go back to conditional formatting here. If you force me, format conditional and single color, and the color will be, let's say, color, black. No, what? Not this one. Rules. Here. Okay, this is the rule I want to change. Format, color, black. Okay.
Okay.
And another rule for the same places to say B2 to L12. And if the text contain 1, we're going to make it white with text color white. Done. Fine.
Okay, fine. Thank you.
There may be a way to do it like a plot, but to me, the conditional format is more natural. I'm just changing the way to show the numbers, but these are the numbers. Inside you have ones and zeros. And yes, no?
Yes. Okay, I got it.
Even more square. So here you can play with other texts.
Yeah, we can try your examples to see.
Yeah, you can try. Yeah. So I think I published the sequence. I am going to fix the links that are broken and see if I can make it correct next time. But next time we're going to see the same idea and this will soon get us to BLAST. The road here is we go to we have we show global and semi-global alignment. We are going to do local alignment. We are going to do blast, and then we are going to speak about multiple alignment. Okay, that is how we go.
Okay, thank you for this week's class.
Thank you for coming to my class.
I have a question.
Ask questions.
Is there any problem with me, or didn't you upload your web page?
I don't know if I upload my web page or not, because I upload.
I only see the first class.
Are you looking in drylab.org or in the old website?
The one with Andres 11.
No, that I not being updated.
Okay, so you are uploading DryLab?
DryLab, yeah.
Okay.
I will try to synchronize them so they are coherent, but the official is DryLab. I have something else. Whether I will send you an email. There is a paper that I would like you to read. I will send you. Put it here. No, I don't have. Okay, I don't have it. But I will send you a paper to read before. Okay.
Okay.
Thank you for your questions. Every question was useful and enlightening. So we appreciate your.
Thank you for your answers.
Yeah, well, that is my job. They pay me for doing that. Fine.
Do microarray chips work in that way?
It's a little more complicated. The microarray chips work with essentially by hybridization. And hybridization is a little more complicated. Let me talk about that kind of in 3 classes, okay? We are going to talk about primer design, and primer design is also about hybridization. Primer design and microarray design is kind of the same chemistry, kind of. So, but we are going to go with this idea is going to be extended to that. Okay, this is our starting point, and we are going to do more.
Okay.
Good question. Good question. Yep.
I think I didn't understand why the external gaps and internal gaps caused by different reasons, but I think I will need to watch your video again.
Yeah, you can ask with, but if you want to ask a question, ask the question.
I will try to learn myself.
No, wait, you don't ask. Be brave, Asli. Be brave. Ask question. You are brave to ask that I want to ask a question, but okay. There is a kind of a philosophical thing and a practical thing. Philosophically, you can just decide randomly, like, look, I don't care about the gaps outside. It's my decision. I don't care. I only care the gaps inside the sequence. I don't care the gap outside. That is a kind of random decision, but who cares? I make a decision. But there is a biological reason, or let's say scientific reason to choose that. And the reason is because we distinguish the information that is missing because we don't measure it. Some part of the DNA we have not seen, but exists, but we don't know, versus part of DNA that we know and we know is different. When you compare two things, you can say either they are equal, they are different, or we don't know. Okay, and we don't know is essentially because we lack information. So that is kind of the basic point here. When you have two sequences, you can compare and you can say they are equal or they are different. When you don't know, you cannot say that they are different because you don't know. So in that case, what we are doing is that we say we don't punish, there is no cost for the gaps outside because that is the part that we don't know. Okay?
Okay.
That is why we call external gaps means the part that we don't know.
Okay.
Okay. We are going to see it then again anyway. But at some level, it's also kind of arbitrary. I can decide, look, in this kind of problem, I want to ignore this case. I want to focus here. Because remember, at the end, we have to define, we are kind of trying to make a model of a biological problem, and we want to make a model that represent what is important for biologists. Okay. So at the end, the decisions are based on biology. Fine.
Yes.
Tamam.